Welcome students. Now we are dealing with the chapter Mineral Nutrition. In this chapter we are coming to this section, critical concentration. What is critical concentration? We know that for every organism, plants as well as animals, anything if we take more than our need, it may become toxic. So a concentration at which 10% of dry weight of tissue is reduced is considered as toxic. So a concentration at which 10% of dry weight is reduced, the dry weight of tissue is reduced is considered as toxic. So to increase the productivity and also to avoid mineral toxicity, knowledge of critical concentration is essential. So we must know the critical concentration of each mineral nutrients or each elements to mineral nutrients, that how much the plant required for that metabolic process, the critical concentration of that particular element. So mineral nutrients lesser than critical concentration, it causes deficiency symptoms. If the nutrient lesser than critical concentration, it show deficiency symptoms, whereas mineral nutrients that is more than normal concentration, it's called toxicity. So nutrient that is less than normal concentration, it shows deficiency. Nutrients more than concentration, more than normal concentration, it becomes toxic. So nutrients which is lesser than concentration, it causes deficiency symptoms. Nutrients which is more than normal concentration, it becomes toxic. So here is the graph of critical concentration. We are plotting the concentration of nutrition in plant tissues versus growth as a percentage of maximum range. So the mineral nutrients lower than the normal concentration, it show deficiency symptoms, whereas it is higher than the normal concentration, it becomes toxic. So this is the advocate sum. This is the advocate sum. So this is the critical concentration of every particular elements. So above the normal concentration, it become the mineral nutrients become toxic, and lower than the normal concentration, it show deficiency symptoms. Okay, now we can go to the topic mineral toxicity. Mineral toxicity. So any mineral nutrients that is more than the normal concentration, they causes toxicity. First one, manganese toxicity. Manganese toxicity. The increased concentration of manganese, that is higher than the normal concentration, will prevent the uptake of iron and magnesium. So the increased concentration of magnesium may causes the prevention of uptake of iron and calcium. Iron and magnesium. Also, it affects It affects calcium also. That is, it prevents the translocation of calcium. To the shoot apex. So, the increased concentration of magnesium may cause the prevention of uptake of iron and magnesium, where like that prevention of the translocation of calcium to the shoot apex. The symptoms of magnesium toxicity are appearance of brown spot surrounded by chlorotic veins. That is, the appearance of brown spots on the leaf veins. The appearance of brown spots on the leaf veins are the symptoms of manganese toxicity. So, manganese toxicity is the increased concentration of magnesium may cause the prevention of uptake of iron and magnesium, also the prevention of translocation of calcium to the shoot apex. Now, aluminium toxicity. Aluminium toxicity. Aluminium toxicity, what it causes? It causes the precipitation of nucleic acid, inhibition of ATPase, inhibition of cell division and binding of plasma membrane with galvanizing. So, aluminium toxicity causes precipitation of nucleic acid, inhibition of cell division and inhibition of ATPase enzyme and binding of plasma membrane with calmodulin. Inhibition of cell division and ATPase enzyme. Then also it binding of plasma membrane with calmodulin. Calmodulin is a calcium binding protein. So, aluminium toxicity may cause the precipitation of nucleic acid, inhibition of cell division and ATPase enzyme, binding of plasma membrane with calmodulin. Now, iron toxicity. Iron toxicity. Actually, iron and manganese exhibit competitive behavior. Iron and manganese exhibit competitive behavior. The increased concentration of iron may affect the uptake of or the absorption of manganese. So, iron toxicity will affect the absorption of manganese. The possible reason for iron toxicity is excess usage of chelated iron in addition with increased acidity of soil. So, the iron and manganese toxicity can be solved by using fertilizer with balanced ratio of iron and manganese. So, we can solve the issues by using fertilizers having same ratio of iron and manganese. So, these are about mineral toxicity. There are three types: manganese toxicity, aluminium toxicity, and iron manganese toxicity. Now we are moving to hydroponics and aeroponics. First one, hydroponics. What is hydroponics? It is simply but soilless culture. Soilless culture. In hydroponics, what is that? The plants are grown in nutrient solution. It's not, normally the plants are grown in soil, but in hydroponics, the plants are grown in nutrient solution. Actually, the techniques was invented by Bonsack. He developed that a method of growing plants in nutrient solution. In this technique, roots are immersed in the nutrient solution. Roots immersed in nutrient solution, and air is given by using a tube. Air is provided or supplied with the help of a tube. So in hydroponics, soilless culture, Bonsack developed this technique. In hydroponics, the roots are immersed in solution containing nutrient and the air is supplied with the help of a tube. Second one is aeroponics. Second one is aeroponics. Actually, Soyford Hill and David Turger those scientists jointly developed this technique. Actually, in this technique, roots are suspended in air and the nutrient solution or nutrients are sprayed over the roots with the help of a motor. In aeroponics, the roots are suspended in air. Roots are suspended in air and the nutrient solution which is given with the help of a motor drawn rotor. Motor drawn rotor. So these are the two techniques of culturing of plants without the help of soil. First one is hydroponics and the second one is aeroponics. Now we are going to discuss detailedly about nitrogen fixation. So what is nitrogen fixation? We know that life on earth depends on nitrogen cycle. Life on the earth depends on nitrogen cycle. In the atmosphere, the nitrogen is in the form of N2. That is 2 nitrogen that is bonded with the strong bond, triple bond. So nitrogen is available in atmosphere in the form of N2. It is highly strong in nature. So the process of converting atmospheric nitrogen into ammonia. The process of converting atmospheric nitrogen into ammonia is called nitrogen fixation. Nitrogen fixation. So the nitrogen fixation can be done or occur by two methods: biological nitrogen fixation and so nitrogen fixation are of two nitrogen fixation are of two non-biological nitrogen fixation and biological nitrogen fixation. In biological nitrogen fixation, it is again classified into non-symbiotic and symbiotic. And symbiotic. And in symbiotic, it is again classified into non-legume and legume. In non-biological classification, non-biological nitrogen fixation, it is industrial and lightning. So, these are simply nitrogen fixation. Nitrogen fixation is the process by which the atmospheric nitrogen is converted into ammonia form. Nitrogen fixation are of two types: non-biological and biological. Non-biological nitrogen fixation are include industrial fixation and thunder and lightning, whereas biological nitrogen fixation are of two types: non-symbiotic nitrogen fixation and symbiotic nitrogen fixation. In symbiotic nitrogen fixation, it's of two types: non-leguminous plants, also leguminous plants. Now we can go to the detailed non-biological nitrogen fixation. Non-biological nitrogen fixation. Non-biological nitrogen fixation. First one, industrial. Industrial is nothing but the conversion of atmospheric nitrogen into ammonia. The industrial nitrogen fixation is called Haber-Bosch process. N2 plus 3H2 gives 2NH3. That is the atmospheric nitrogen which is converted to ammonia. This is called Haber-Bosch process. And the second non-biological nitrogen fixation is lightning. So, during thundering and lightning, the natural electrical discharges fixes the atmospheric nitrogen. The natural electrical discharges fixes the atmospheric nitrogen. So these are non-biological fixation. It's of two types: industrial and lightning. Industrial process is called Haber-Bosch process, and lightning is called natural electric discharge fixes the atmospheric nitrogen. Now we can move to biological nitrogen fixation. In biological nitrogen nitrogen fixation. The nitrogen fixation can be done with the help of nodulation, without the help of nodulation. So, first one, nitrogen fixation with nodulation or nodule formation. Second one, nitrogen fixation without nodulation. Okay, so biological nitrogen fixation can be again classified into nitrogen fixation with nodulation and nitrogen fixation without nodulation. Now first we can detailing look what is nitrogen fixation with nodule formation or nodulation. Rhizobium is a bacteria. Rhizobium is a bacterium that is found in the leguminous plant. Leguminous plants are plants that included in the family Leguminaceae. The old name of Leguminaceae family is called Fabaceae. So Leguminaceae family members plants containing Rhizobium bacterium, and these Rhizobium bacterium fixes nitrogen. How they are fixing nitrogen is they produces a structure, or Rhizobium produces nodules, root nodules. The symbiotic association between the Rhizobium and the plant shows the formation of root nodules. So, the symbiotic association Rhizobium with the plant shows a symbiotic association. Symbiotic association means which is favorable to both bacterium and plants. That is, host and bacteria get equal benefits. So, the symbiotic association between Rhizobium and legumes is the reason for the formation of root nodules. That's why it is called as nitrogen fixation with nodulation or nodule formation. So, root nodules are formed due to the bacterial infection. Rhizobium, then how this Rhizobium fixes? Rhizobium enters into the root cells or the host cell and it divides. It remains separated from the host cytoplasm by a membrane. What are the stages of root nodule formation? First one, the legume plants. Actually, the Rhizobium is somewhere in the soil, and the plant, this is the plant. So this leguminous plant, the root cells may produce some secretion. Root cells may produce some secretion which will attract the Rhizobium. Attract the bacterium Rhizobium. So the leguminous plants root cells may produce some secretions which may attract the Rhizobium bacteria. This Rhizobium reaches the rhizosphere. So rhizosphere is nothing but the roots inside the soil. This area is called the rhizosphere. So the Rhizobium reaches the rhizosphere and enter into the root hair, infects the hair and leads to curling of root hairs. So what they done is, the Rhizobium affect the root cell. Rhizobium affect the root cell and it causes the curling of root hairs. Curling of root hairs. It affects the curling of, if this is a root hair, it may causes the curling of the root hairs due to effect of this rhizobium bacteria. This rhizobium bacteria may enter into the root cell and it may affect the curling of root hairs. The infection thread grows inwards and separates the infected tissues from normal tissue. What they done is, from this rhizobium, the infection thread may originate, and what they done is they separates the infected tissues from normal tissue. So the infection thread grows inwards and separates the infected tissues from normal tissue. A membrane-bound bacterium is formed inside the nodules. So when the bacterium affect the root hairs and they enter into the roots and they forms what it is, the bacterium forms its own membrane. If you consider this is the root cell, bacterium is enter into the root cell and it produces its own membrane. So this is called bacterioid. So a membrane bounded bacterium is formed inside the nodule. Is called bacterioid. Cytokinin from bacteria and auxin from host plant promote cell division and leads to nodule formation. Actually, this is the root hair. In the root hair, you can see some nodules. So this is the root hair. This is the nodules. So inside the nodules, how this nodules is formed that cytokinin and auxin are the two hormones which is responsible for the formation of auxin by cell division. Cytokinin from bacteria. So rhizobium provides cytokinin and root hairs contain auxin. So plants provide auxin. So this auxin and cytokinin, they jointly promote cell division. So this cell division may result in the formation of root nodules. So these are the stages of nodule formation. These are the stages of nodule formation. Inside these nodules, rhizobium forms a bacterioid structure and that bacterioid structure may convert the atmospheric nitrogen into ammonia form or plant absorbable form. This is the leguminous plant case. Now we can look the non-legume. Non-legume. Alnus and Casuarina are plants without legumes contain a bacteria called Frankia. Frankia is a bacteria found in Alnus and Casuarina plants, and Frankia fixes the atmospheric nitrogen. Then Klebsiella. Klebsiella is a bacterium that is found in Cypripedium. Klebsiella is a bacterium which is found in Cypripedium and it also fixes the atmospheric nitrogen. So these are about the nitrogen fixation with nodule formation. Now we can look nitrogen fixation without nodule formation. Nitrogen fixation without nodule formation. Several plants and prokaryotes are involved in nitrogen fixation, that we already know. So there are some plants and prokaryotes. First one is lichen. We know lichen is the symbiotic association of algae and fungi. Lichen is a symbiotic association of algae and fungi. In lichen, Anabina and Nostoc, that is the algal part, Anabina and Nostoc fixes the atmospheric nitrogen. Anabina and Nostoc fixes the atmospheric nitrogen. Second one is Anthocellose. Anthocellose is a bryophytes. Inside the Anthocellose, the blue-green algae or cyanobacteria called Nostoc is responsible for the fixation of atmospheric nitrogen. Then third one, Azolla. We know that Azolla is a pteridophytes. It containing Anabina azolla, containing a blue-green algae, Azolla containing a blue-green algae called Anabina azolla. Anabina azolla. Then Cycas. Cycas is a gymnosperm. Cycas having a root called coralloid root. Inside that coralloid root, symbiotic association is occurring with Anabina and Nostoc. So these are some plants and prokaryotes that is responsible for the fixation of atmospheric nitrogen. So these are about symbiotic nitrogen fixation. So in symbiotic nitrogen fixation, we study nitrogen fixation with nodule formation, nitrogen fixation without nodule formation. Now we are going to the non-symbiotic nitrogen fixation. Non-symbiotic nitrogen fixation. That is, free-living bacteria and fungi also fixes nitrogen bacteria. Some free-living bacteria and fungi fixes atmospheric nitrogen into plants' absorbable form of nitrogen. Some bacteria are, first one, aerobic bacteria. Aerobic bacteria like aerobic bacteria like Azotobacter, Regenica and Dersia. Those bacteria helps in aerobic nitrogen fixation, whereas in anaerobic Azotobacter, Regenica and Dersia, whereas in anaerobic Clostridium. Whereas in photosynthetic it is Chlorobium. Then chemosynthetic bacteria like Disulfovibrio. Then free-living fungi like yeast and Polylaria, free-living fungi. Yeast and Polylaria. Then last, cyanobacteria. We already found so many cyanobacteria like nostoc, anabina, etc. So cyanobacteria include nostoc, anabina and Oscillataria. Nostoc, anabina and Oscillataria. So these are about non-symbiotic nitrogen fixation. So we found nitrogen fixation are of two types, biological nitrogen fixation and non-biological nitrogen fixation. Non-biological nitrogen fixation include industrial and lighting, whereas biological nitrogen fixation include symbiotic nitrogen fixation and non-symbiotic nitrogen fixation. In symbiotic nitrogen fixation, it is of liquid or and non-liquid. So these are about nitrogen fixation.